Yes, sir. Yes, sir. Please, 1 minute wait.
Okay, no problem.
Yes, sir. Yes, sir. Please, 1 minute wait.
Okay, no problem.
శ్రావణి అన్మ్యూట్ చేసుకోమను.
Sir. Good afternoon, everyone. Welcome to today's session. I'm honored to introduce Professor Veerabrahma KISHAN Garu for online 6-day faculty development program. Sir has completed his bachelor degree of pharmacy from Kakatiya University with first rank and also gold medal. Sir has completed his master's degree of pharmacy from Andhra University with distinction, and Sir has got his PhD from Friedrich Alexander University, Germany. Sir has postdoctoral experiences from Friedrich Alexander University, Germany, and also University of Göttingen, Germany. Sir started his career as lecturer in Department of Pharmacy, KU, for almost 8 years, and Sir has DAAD Fellow of Friedrich Alexander University, Germany, for 3 years, and Sir was worked as Associate Professor of Pharmacy nearly 12 years, and Sir is currently working as Professor of Pharmacy, KU, from 16 years. And Sir has awarded with Srimati Arikapudi Lakshmi Rajam Memorial Gold Medal by Kakatiya University, and Kishan Garu also awarded with Gold Medal from Indian Pharmaceutical Association, Hyderabad. And Sir is also having the award of Pharmacy Teacher of the Year award by Association of the Pharmaceutical Sciences and Nanotechnology. And Sir has received Telangana State Memorial Award from the Government of Telangana. Sir is also a receiver of Dr. Manjushri Patla Memorial Best Pharmaceutical Scientist Award from the Association of Pharmaceutical Teachers of India. Sir is also a receiver of Lifetime Achievement Award from APP. And Kishan Garu has honored with the certificate of APP from UCPS, KU, and also PCI New Delhi, and then Kakatiya University. Sir is also having 3 fellowships. Sir has 37 years of research experience. SAR has guided 15 PhD scholars, and SAR also guided 106 pharmacy students, that is MPharm students, and SAR has received 85 lakh rupees nearly for research project grants. SAR has established research collaboration with Professor Axel Zach and from the University of Germany, and also from same university with Latesh, and also with the research collaboration of Dr. Y. Venkateshwarulu, scientist from IICT Hyderabad, and also having the collaboration with Dr. P. Uday Kumar, Director of NIN Hyderabad. And as a resource person, SAR has delivered 50 lectures. SAR worked as an affiliation committee member, and SAR is having 72 research publications. SAR has delivered 24 lectures at international conferences. Once again, I would like to welcome the Professor KISHAN Garu to the today's session.
Sir, unmute చేసుకోండి sir. Sir, please unmute yourself. Sir. Sir, click చేయండి sir అక్కడ. Okay, sir. Okay, sir. Okay, sir.
Okay.
Yes.
Now, are you able to listen to me?
Yes, sir. Yes.
Good afternoon, ladies and gentlemen. And I'm really sorry for being late in starting the session. And in this session, Professor Gopinath, the principal of the college, who requested me to address this particular faculty development program being sponsored by All India Council for Technical Education. The title of this particular program is on pharmacy education and research, present and future challenges. It's really an interesting topic, and the current scenario in pharmaceutical field is that because of the increased population, variety of diseases and disorders, there is a big challenge for the discovery of new drugs, new methods, and also other variety of reasons are there for improved requirement. And here all of us are belonging to the category of pharmacy, and more so as pharmacy teachers and pharmacy students also, because a teacher is always a student, then only he can learn and he can teach. So in the pharmacy curriculum, new topics, new inventions, new developments have to be inserted. Then only the teachers will be able to deliver the updated knowledge to the students, and the purpose of this is nothing but the same. And I'm as a matter of fact, all of you are basically working as teachers, and I'm also a teacher, and I'm not really going to teach you anything. It is a kind of sharing of my experiences and knowledge in the area of vaccine drug delivery systems or vaccine delivery systems. Although many of us have taught this topic for the BPharm students, and even MPharm also, some of you might be, but still I try my best to give some inputs in the form of an overview in this particular lecture. The lecture is somewhat delayed, but still I try to give some information so that you can just recollect whatever that you have learned previously, and also some of the young teachers can also concentrate a little more in this particular area, so that you can teach to your students in a better fashion. That's the purpose for which I have taken this particular job when Dr. Gopinath asked me to deliver a lecture in this area. And I also thank you for your interest in listening to this lecture. And I also thank Professor Gopinath for giving me an opportunity to address to this online Zoom meeting to all the teacher colleagues working in different parts of the country. And now, as just now I told you that it is the topic is vaccine delivery system, and this particular topic is having the following contents. Basically, it's an overview. Just now I told you that a brief introduction, historical account also I will give you, and then the current challenges of vaccines, and to overcome these challenges, a variety of things are done, that is in the form of vaccine delivery systems. And here I'm going to give you some idea regarding what is single shot vaccine, mucosal vaccines, DNA-based vaccines, and similarly current scenario is full of COVID, and what type of developments are there in this COVID vaccination program also I'm going to give you some idea. And now first we will go to the slide, next slide. Basically, we all know that health is wealth. If you are healthy, you are wealthy. That was the old proverb or adage we believe in. And the basis for this is is that normally human beings during their survival, they are supposed to be infected with some disease, maybe caused by bacteria or viruses, etc. So this is always be there in the life of any person or any living being. So naturally, so here, whenever there is possible problem of infection, so there are ways of treating. That is, you can treat them, or otherwise you can prevent them. So the vaccines are basically dependent on the prevention. But here the thing is, prevention is better than a cure, or an ounce of prevention is worth a pound of cure. That is Benjamin Franklin said some time back. So that means prevention is always a good thing. You can avoid a lot of loss. Financially, you can also minimize the costs, and you can maintain good health. So here, basically, the vaccines are designed, are intended to prevent infections and to reduce the cost of maintaining the good health. And here the vaccines are available for human beings, not only human beings, even for the animals and the poultry birds, because this is a vaccine industry is a very, very big industry. And this is available, but the strategies and the developmental things are equally valid for all these category of living beings, that is human beings, animals, and poultry birds. So vaccines are actually, if you see, they are active immunizing agents. They are not passive immunizing agents, but they are active immunizing agents, and they contain what's it called? Sorry. They contain the infectious antigens which are there, and they try to create or stimulate the immune system to produce the antibodies. And these antibodies are going to prevent the infection. So here, if you just look into the history, the word vaccine is actually originated from the cowpox virus, known as the vaccinia virus. Vaccine means it's a cow in the Latin language. And here, there are variety of communicable diseases have been noticed, like tuberculosis, cholera, typhus, plague, polio, smallpox, etc. were very, very dreadful diseases in the history of mankind. And a large number of people used to die in the different epidemics that were there. And sometimes even they spreaded like pandemics also. And recently now we are having what is known as COVID-19 is a pandemic now we are facing it. And here, just this is a small picture where the first vaccination protocol was undertaken by Edward Jenner in 1796, first vaccination. And he inoculated to a small 8-year-old boy known as James Phipps, and he was protected against the smallpox. And this smallpox vaccination involved the application of cowpox material onto the boy. So that was the first attempt, recorded as attempt for the vaccination. And next, what you will see in this picture is Louis Pasteur. You know that is a say bacteriologist, and he developed a vaccine against the anthrax. So this is he developed from anthrax bacilli. So that is one vaccine first developed, and this developed vaccine is called the attenuated vaccine. And next, subsequently, live vaccines were also developed, and these vaccines particularly developed in the case of rabies. And here what exactly they have done is in 1884 to 85 period, Louis Pasteur and along with his co-workers developed some vaccine against the rabies. So the rabies is a disease which is whenever the dogs are bite biting the particular individuals, or the dog bite, let us say, this will spread. So to against to get this kind of exposure, rabies vaccines have been developed, and they have been inoculated in the abdominal region. And here this was actually developed from sources like sheep and rabbit. That was historical. Later on, cell culture based were also developed. So here, and that was, the virus was having some ability to further propagate in the body, whereas some other vaccines were also developed, known as inactivated vaccines, and these vaccines are whole cell bacterial vaccines. In 1896, Wilhelm Kohlhe developed heat-killed bacterial vaccine, particularly cholera vaccine, and which was given by parental means. In 1896, Research, Pfeiffer and Almorth Wright, they have developed independently typhoid vaccine. Similarly, Waldmer Hofkin, 1911, he developed another vaccine. So like that, there are so many people have contributed for the vaccines. And one important point is that just now we have talked that vaccines are given as a prophylactic or to prevent the infection. The unique situation in the case of rabies is that after the infection, that means whenever the dog bite was there, subsequent to the dog bite, the immunization was done. That is called the post-exposure. So that was there, and usually the old practice was that the animals like rabbit or sheep, etc., were inoculated with the virus, and the animals were killed, and then spinal cords were separated, and the spinal cords were dried for, say, maximum 14 days, then 13 days, 12 days, like that. And subsequently what they do is they prepare suspensions, and these suspensions are given to the individuals one after the other. That is starting from 14th day dried suspension, then 13th day, like that. The virulence or the potential of that particular vaccine will be very, very negligible, and then the person gets the required immunity. This is a very controversial procedure, as a matter of fact, but it was practiced. And then now, after listening to this particular few interesting, say, vaccinations which you have, that is vaccines, I briefly tell you what exactly this vaccine is going to do. So whenever we are going to immunize a particular person, these vaccines are expected to provide immunity, and this immunity is of two types. One is called humoral immunity, and the other one is cell-mediated immunity. Here, what is humoral immunity is nothing but production of antibodies. So here, the B lymphocytes actually produce the, say, antibodies, and this is called what is known as the humoral immunity. And then another immunity is called the cell-mediated immunity is there, and this particular immunity involves that a particular cell known as the T lymphocytes, cytotoxic lymphocytes, that means these cells are capable of killing those cells, and those cells which are infected with particular virus. That means, so virus propagation can be reduced. That means an infected cell is there in the life cycle of virus propagation. First, they attack the cells, and these cells will provide environment for the growth and multiplication of the viruses. And in that stage, the cell is killed. This means the viruses are also killed. No further propagation. That is one way. Or otherwise, antibodies are produced, and antibodies will attack on the virus so that they are neutralized. So these two, generally the immunizations will be there, that is cell mediated as well as the humoral immunity, and the vaccines are expected to do this kind of jobs. And here, if you look into the say currently existing traditional vaccines, there are variety of traditional vaccines are there for both viral as well as for the bacterial for human application. So here, live attenuated means they are living, they are not completely dead, and inactivated means they are not going to be living, and they are whole virion, or that means whole, or sometimes split, or means some portions, and sometimes you can also get combinations. So variety of ways of handling of the vaccines are there. So examples you can say, live attenuated means vaccinia, yellow fever virus, oral polio. You know, oral polio is a very famous one, like that. And inactivated means influenza virus. Rabies is also inactivated virus is there, and then here inactivated polio virus is also there. And other examples are many. Influenza, hepatitis B, these are called as subunit vaccines. And measles, mumps, and rubella, these are all MMR vaccines, are the combination vaccines. And there are so many vaccines are also added. And now we'll see here bacterial vaccines. This is bacterial vaccines, and live attenuated is there, inactivated whole organism, toxoids, subunits, combination in all these cases. So BCG is a very one, Bacillus calmette-guerin, Salmonella typhi TY21. This live attenuated one, Bordetella pertussis, cholera, vibri, and then typhi, the Salmonella typhi, etc. These are inactivated. Clostridium, tetanus, and Corynebacterium, diphtheria, the diphtheria toxoids, they are called as tetanus toxoid. So these are toxoids. And then subunit vaccines are also developed for certain, say, vaccines like Haemophilus influenza, Neisseria, etc. And the combinations are also there. Like, for example, you must have had a DPT, that is diphtheria, pertussis, and tetanus. So here, this type of things are there. So this is all a little history and the existing traditional both bacteria and what's it called, viral vaccines. So now what are the actual challenges? So here the challenges for the existing vaccines is an important thing. So almost all the vaccines are injected. Injected means they are single dose may be there, sometimes 2 or 3, etc. That means if you see the universal immunization protocol schedule, there are schedules are there, and you will be able to see the number of injections that are to be in a childhood. Large number of infections are there, and they are going to be prevented. So a child is exposed to many times the pricking or the injections. So it's a very painful, and they try to escape, and it is rather inconvenient also. Pain is there, sometimes fever. Patients have high dropout rate. That means if the dose prescribes that 2 or 3 doses are there, sometimes they may not take. So as a result, the dropout rate and unstable. Usually they must be stored in refrigerated conditions, and somewhat expensive also. And the other side effects are also there for the existing vaccines. So now you have to, because of the advances in the knowledge of the science, biosciences or the life sciences, you are expected to provide a better and safe, highly immunizable or immunity power say antigens. So now I'm showing a picture here. See, throughout the globe, say any child, whether it's an American child or European child or African child, etc., whenever they are exposed to vaccination, they don't like, they start crying, they try to escape. This is a simple picture. So now I told you that there are some challenges are there, and the new vaccine delivery systems are required. The WHO, because here most of the times the infectious diseases are prevented by immunizations. World Health Organization is one organization of the UNO. This organization promotes the immunization. That means preventing the infection, a spread of infection, and the epidemics, pandemics, etc. The job is to, and they try to provide some money to the different companies, and so that the companies will prepare the vaccines required, and they will be distributed at a subsidized prices or even free also based on the requirement. And the governments will usually take it as a, not as a regular business, the companies. So here the WHO is encouraging researchers to develop what is known as single administration or one-shot parental vaccine. That means if a particular vaccine is supposed to be taken as 3 doses, if by any reason, if one vaccine dose is sufficient to meet the 3 doses at 3 different intervals, that is considered to be an advancement. So that is single administration and easy to deliver. So most of the existing one are parental injections. By any reason, if you can develop the same injection in the form of a orally, because the oral administration is easy and you don't need any kind of support from the skilled person. The skilled person means nurse or somebody must inject. So that is not required. You can, a mother can give to the baby. So like that. And similarly, transdermal vaccinations are also there. That means under the skin, if you are going to put your vaccine without any pain, invasiveness is not there. So that's also encouraged. And thermostable, because cold chain is required for normal maintenance. Thermostable means if you can put just like a tablet in the room temperature, that is a better one. And they must be safe, and the efficacy of that particular vaccine should be high, and rather they should be cheap. So these are all some of the areas of the research where the WHO encourages the companies or the scientists to undertake the research. So here, really interested to develop a vaccine, so you should know what exactly the components of the vaccine. So basic vaccine consists of an antigen. Antigen is nothing but a foreign material which our body considers a foreign material, and against which we try to produce antibodies. And the foreign material may again enter into our body by what means in the form of an infectious organism like bacteria or viruses usually. So these are the 2 ways of entering naturally. So naturally our body will try to respond by producing the corresponding antibodies, etc. So here that is an antigen. That portion is called the antigen in the vaccine. Next, so to give a formulation, to give a better efficacy for the immunity, certain other components are also included in the vaccine development. So one among them is called the adjuvants. And so these adjuvants are actually the agents that can induce the immunity, or they can strengthen, that means they enhance the immunity. Normal vaccines will produce some immunity, but the power of immunity is not that high. So naturally you can increase the strength of the vaccine by inclusion of what is known as adjuvants. And these adjuvants will act as how they try to stimulate and proliferate and then differentiation of the immunocompetent cells, and these are nothing but the B and T cells. That is, just now I told you that B cells will produce the antibodies, and the T cells will be acting on killing of the, say, infectious cells. So these two types of immunities are generally there, and these adjuvants will try to promote this particular activity. So here advantages of vaccine adjuvants are they increase the potency of weak and small synthetic peptides which are there, enhance the speed, vigor, and other say persistence of immunity responses, increase immune response in immunologically immature people, old people, senescent, etc. And sometimes they try to modulate the humoral response or the cell-mediated response by, say, working through MSC class 1 and class 2 responses. Similarly, they will increase the avidity and the specificity, quantity, isotype, variety of say characters are there for the immunological responses, and they are all going to be increased. And the quantity of the antigen that is required to give the good immunity can also be reduced, or sometimes the antigen can be given along with some other things also. So these are all some of the advantages of the this adjuvants, and they usually have some mechanism. Antigens are brought into a proper microenvironment. That means after giving injection, it requires a specific environment. Into that, it is going to be transported or taken. So that is one microenvironment. And then gradually it tries to release the antigen. So it's not like that sustained action type. For example, if you take antigen, it should slowly release, not in very fast. That is not good for the immunological reactions. And when you are releasing, it provides some environment wherein the macrophages, which are naturally existing cells in our body, we're working in the defense mechanism, and also some lymphoid cells, these are all going to be recruited and activated. And this APC is nothing but the antigen presenting cells like dendritic cells, etc. are there. And here they also sometimes complements and then synthesize and the secretion and variety of say immunologically relevant say cytokines. So these are all the things which are going to be activated by the adjuvants. And here particularly they try to provide an opportunity for the T cell epitopes to the MSC class 1, and here CD8 plus cells, CD4 plus cells are going to work in the production of antibodies. So here this, and there are some risks are also like, for example, adjuvants can cause some kind of inflammations are there, that is very strong inflammation and adenopathy, some headaches, anaphylaxis, and variety of teratogenesis, abortogenesis, carcinogenesis, and variety some other side effects are also there. So that means in the list of adjuvants, there are variety of adjuvants have been tried by scientists, and naturally you can choose based on your experience and then the results of the particular studies, and so that you can pick up. So here some of the adjuvants which are tried include alum or aluminum. Aluminum and calcium salts were there. One adjuvant was a complete France adjuvant, which is not usually given for the human beings. Trehel is only for the research purposes they are used generally. And trehalose dimycolate, muramyl dipeptides and tripeptides, dextran, monophosphoryllipid A, and immunostimulating complexes are also tried as adjuvants. So now, the Food and Drug Administration has approved these aluminum salts like alum, aluminum hydroxide, etc. But usually this is not suitable. Most of the vaccines will contain tetanus toxide. You must have formal toxide. So they will be usually having this. Now they are not suitable for subunit or synthetic vaccines. One important, and they do not trigger the CMI. That is also an important point with aluminum salts. And not suitable for mucosal immunity. That means if you are taking through the oral route, side effects are there. And the first portion of the vaccine is that the antigen, which is important candidate or point. Next thing is adjuvants, which try to trigger the immune response. And then we are having what are known as carriers. So here, this carrier is nothing but an immunogenic protein. Sometimes what happens, the antigen itself is not in a position to. So naturally, it may be tagged to the carrier protein so that the complexity of the antigen is increased and it works as a better antigen. So here, some of portions of the called the haptens, and these are weakly immunogenic in nature antigens, and they themselves do not have the capacity to trigger the immune responses, and they may be linked to others. For example, bacterial toxoids, tetanus, diphtheria toxoid, pseudomonas, meningococcal, outer membrane toxin, cholera toxin, etc. You can put these to the other antigens so that the antigen becomes activated. So even living vectors were also tried as carriers, like vaccinia virus. So normal virus, it is capable of infecting us, and you can use that virus as also carrier for some antigens. Adenoviruses, and similarly salmonella and attenuated vibrio cholera, attenuated shigella. These are all bacterial vectors, viral vectors. We will be talking about these also slightly later. And then vehicles. So carrier is one thing, vehicle is the one thing, adjuvant is one thing, and antigen. So here, these vehicles are not actually having any important role in the immunogenicity of the particular vaccine, but sometimes they may contribute. That is also there, and these also act as a substrate for the adjuvants and the antigens, and they may try to create what is known as carrier complex. Adjuvant along with the antigen, a complex may be generated. They may also produce some antibodies sufficiently. So here in the vehicles, you will see variety of things like normal emulsions, like mineral oil emulsions, vegetable oil emulsions, and squalane and squalene emulsions. There are some companies which have prepared as vehicles. That means you put your antigen in this particular emulsion, it acts as a vaccine because it is a vehicle. So here MF49, Chiron Corporation, Syntax Research, SAF, and liposomes were tried as carriers. Sendai viruses and immunopotentiated reconstituted influenza viruses. These are also sometimes called as virosomes, and biodegradable polymeric microspheres, protein co-creates, and even some scientists are also considering that as edible plants. So these are all the ways of putting the antigen for the immunization purposes. Now, just now I told you that the antigen is, vehicles. Among the vehicles, emulsions are simple. One can prepare it in the university college, in the colleges, pharmacy colleges. An example of a marketed is that SAF, that is Syntax adjuvant formulation, which consists of OW emulsion, 5% squalane, 2.5% pluronic, L121, 0.2% polysorbate, 80 in phosphate buffered saline. So this is a composition, and similarly MF, that is microfluids F9, 59, and it's also OW emulsion, and they may have globule size 200 to 300 size, and the percentage of squalene is 4.3%, 0.5% Phen 80 plus span 85 also is there. So like that, there are different compositions are there. One can prepare the emulsion and you can do that. So here liposomes were, because liposomes you have already studied as what is known as delivery systems for the drugs. So you can also use these same pliable, flexible delivery systems for the delivery of the vaccines also, because the vaccines are containing, if you see the antigen nature, it is a whole bacteria may be there, or whole virus may be there, or a part of the virus. Like for example, toxide is an excreted portion from the bacteria, that is toxin is converted to toxide. So like that, you can make it the vaccine. It's a macromolecule may be there, or even total complete virus or bacteria may be there. Variety of materials are going to be there in the vaccine. So here liposomes, they are fluid, pliable, bilayered with internal aqueous environment. They are usually safe and very effective, and these provide both that is called humoral as well as the cell-mediated immunity. And one important vaccine marketed in Switzerland is by liposomal vaccine for hepatitis A. And the liposomal vaccine was also tried in Japan against what is it called? It contained the portions of heme agglutinin and neuraminidase antigen from influenza. And sometimes liposomes have some problems like oxidation, etc. Some scientists are also have tried with neosomes. The non-annex surfactants were there, and they are more or less similar. So here, just you see this is the picture of liposome. So this liposome is a bilayered structure. This is one layer, and the vaccine candidate or the antigen may be entrapped here. Such kind of vesicles you can put, and these vesicles are fluidic in nature, pliable, bilayered, internal aqueous space for putting. And you can make these but you can collapse these particular liposomes in the form of cochleates. So by what way? When you collapse by treating with calcium chloride, usually this will collapse, and in the form of a jelly roll they become, and this is rigid, somewhat solid bilayers, no internal aqueous. This is not going to be there within this jelly-like structure. The antigen is enclosed, so you can use, and these cochleates are somewhat better when compared to the liposomes. So that is, this particular picture shows you how the liposomes and cochleates can be prepared. The liposomes containing inside the material, and here the proteins can be there. If an antigen is protein, you can put peptide, also you can put DNA, also you can put antigens, you can put even drugs, also you can put. This is how they are going to be, say, prepared in the form of a dispersion, and this dispersion consisting of lipids, proteins, and the dispersion is having some amount of surfactant solution or the detergent solution. And if you prepare the subject the same to dialysis technique, and later on you expose this dialysis, means you will be getting the vesicular structures, and further if you add calcium chloride and the water is removed and it is collapsed, and they will roll back in the form of cochleates. Sometimes proteins can be kept, DNA can be kept, or drugs, etc. Whatever the things that you have taken here, they are all inclusive. So this type of cochleates are also important, and people have tried by this. And just now the cochleates, Dr. Cochleates, basically they are very stable phospholipid calcium precipitates, and I have shown the structure, and the unit structure provides large continuous solid lipid layer and rolled in the form of a spiral with no internal aqueous phase. And this is important, and it provides a very good immunity, and you can use them for intranasal purposes through this nasal route, intramuscular or oral route you can use, and even sometimes DNA vaccine, because in this cochleates you can use anyone. So this is one thing. And now specific vaccine delivery systems were also developed. Here one important one is known as a proteosome. Proteosome, it is a vesicle, but it is containing proteins, and these proteins are actually outer membrane proteins from the meningococci. You know that meningococcus, our brain, is going to be infected with this meningococci, and you can take outer membrane protein, and this membrane protein will be utilized in the preparation of this vesicle, and that vesicle is called the proteosome. And the vesicles are around 60 to 100 nanometers in size, and you can put antigens, and these antigens usually having amphiphiliicity, that is both hydrophobic domains and hydrophilic domains, and these two domains are there, and they try to form some type of complexes with proteosomes, and you can put them, you can administer them, and better uptake will be there, better efficacy of the antigen is there in the proteosomes. Some antigens were tried by this particular scientist as a better delivery system, and here these proteosomes can be given through the nasal route or oral route or by means of injection. So this some studies like Shigella LPS vaccine was in the clinical trials, and HIV, that is you know that AIDS virus or HIV, human immunodeficiency virus, HIV envelope protein GP, that is recombinant GP160, was complex to proteasome, and it was studied in mice as well as the rabbit models. Staphylococcus enterotoxin was also used. So like that, there are variety of studies were performed. And then another vaccine delivery system is called the virosome. So what is this virosome? Virosome is nothing but, it is in short, it is written as IRIV, otherwise known as the immunopotentiating reconstituted influenza virosomal vaccine delivery system. It is called virosome. That means it consists of influenza virus portions, some portion, it is reconstituted. So usually they are spherical, unilamellar in nature, as much as size 150 nanometers, developed to potentiate the immune responses against the antigens like glycoproteins, peptides, macromolecular structures. These are all the things which you can put, provide adjuvanticity without any toxicity, stable for human use. Here, because it consisted of the influenza virosomes, influenza portions. So that is what is important. The glycoproteins, etc., derived from this. So here, if you see the basic structure and function aspects, liposome normally, you know that it's a vesicle. This virosome is also same. There's not, only thing is consists of phosphatidylcholine and phosphatidylethanolamine, this there, and then hemoagglutinin from influenza, which intercalates into the phosphate bilayer and then stabilize the liposome base by preventing fusion of the liposomes. So that's what exactly. Here, the next thing is this is having what is known as potentiation, and this potentiation is due to HA1, a subunit which will try to interact with the sialic acid residues which are present on the surface of macrophages, so that uptake is going to be increased. Our macrophages are nothing but immunocompetent cells, and finally the endocytosis takes place and better uptake. And here, whenever there is an exposure to a pH like 5, so usually the HA2 subunit, because it consists of HA1 and HA2 subunits in this protein hemagglutinin, which is derived from the influenza, this within the cell undergoes some kind of conformational changes and exposing the physiogenic peptide with the endosomal membranes. So that means it will also not only going inside into the cell, but within the inside of the cell also it exposes the required antigen to the development. So these type of things are going to be there, and then MSC class 2 molecules are going to be there, and a better response is going to be developed. So here hepatitis A virus, that is a HAV, and a variety of other applications were there for this. And then we have another say vaccine delivery system known as aquasome. So this aquasome is also a vesicular structure, and it is usually delivering the antigen only. And the advantage is that you can predict the rate of this delivery over a period. So how long and how many days, etc. you can predict. The release is slow, and specific targeting is possible. That means where exactly the antigen is released, you can do. It consisted of 3 layers of self-assembling composition with the ceramic carbon center, the crystalline material particulate at the center, and then coating is there, and these here with glossy cellobias, and then polymer. So here this is having degradable calcium phosphate nanocrystalline coated with this pyridoxal phosphate, and then drug or enzyme or viral membrane can be there. And then usually size is around 30 to 500 nanometers, and when you can disperse in water, it gives you colloidal particles. So the aquasome is something like this. So now 3 layers, 1, 2, 3. So here the viral membrane proteins are outside, and this is another, and these 3 layers are there. And you can put, actually you can also incorporate genes with this. Aquasome is not necessarily meant for vaccines only. Vaccine means it's an antigen. Antigen means macromolecule. It's chemically, if you see, it's a big macromolecule, and the complexity is there. There may be protein may be there, lipoproteins may be there, and say glycoproteins may be there chemically. So that means a big macromolecule can be entrapped in these aquasomes, and you can use them. And here on the core, this is the center, and on the top you will be having membrane proteins, and these membrane proteins are going to be recognized by some specific receptors are there, and usually uptake is going to be increased. So here next thing is another delivery system known as a transversome. So this transversome is also a vesicular system which is specifically developed for the transdermal delivery, that is from the skin surface, and they are very, very pliable, and they have the ability to pass through a fine pore. So usually the size of the vesicle is big, but our skin pores are smaller, but they have the flexibility to go into the inside of the skin very easily. So here what it consists of is lipid surfactant vesicles for transdermal delivery. That is nothing but the transversome. And usually for the topical delivery, you can use biomolecules, big molecules, etc. And the nature, if you see, they are ultra deformable. That means the collapse, the structure is collapsed. That means instead of being a vesicle, it can collapse like this. So, and it's not completely destroyed, but it can collapse. That means the shape is altered in such a way that it can pass through the fine pores. And the pore size is something like 100 nanometers, whereas the size of the globule is as much as 500 nanometers. So like that, and usually through this pore, and due to the water gradients, so they will try to move from outside the skin. That means the transversome vesicle, if you have used for the delivery of the vaccine, the advantage is that it is non-invasive. a child will very easily accept. A lot of research is going on, and some vaccines, even in our college also, we have developed some, not completely developed, but we have done some research work with the transversome, with the hepatitis B, but we have not published that work. And then the advantage is that a better uptake is there, and then the T cell population will be increased, and then antibody productions are there. That is a transversome. And then immunostimulatory complexes, ISCOMs. This is also considered as a delivery system. And these ISCOMs are is like say complex molecules around 40 nanometer size particles, very simple, and consists of an antigen is there, cholesterol and QA and phospholipids. All put together results in a complex. And there was a lot of research conducted. Influenza vaccine is licensed in Sweden in the form of ISCOMs. So some research like oval albumin was put and then tested for the say immunity. And this particular slide shows how you can prepare the ISCOMs. It's a relatively simple preparation. It involves sequential addition of the proteins previously solubilized in non-ionic surfactants or the detergents, supplemented with QA, cholesterol, and phosphatidylcholine are added, and then they are assembled and then subjected to during the dialysis they will be assembled, or sometimes gradient centrifugation also you can do. And then this actually acts as a potentiating agent for humoral and cellular immunity, and usually given by parental route. And not only the thing is, virosomes are there, proteosomes are there, and then aquasomes are there. And just now I told you this transversome. And apart from that, even nanoparticles, microparticles were also tried as a vaccine delivery systems. And this particular dendritic cell is, in this picture is a dendritic cell, and there's a normal cell, and it is immunologically important cell, and it is otherwise known as an antigen presenting cell, which is present in our body, and they uptake the foreign bacteria, foreign particles, which when they are injected into the subcutaneous tissue, etc. So dendritic cells act as a sentries of the body. That means they try to protect our body against the infections from outside, and they actually take the pathogens and present them to the T cells, produce cytokines to direct immunological responses. So nanoparticles for vaccine purposes, what exactly they do is, if you put your vaccine or the antigen in the form of a nanoparticle, this nanoparticle acts something like a pathogen, that is mimicking the pathogen surface characteristics. That is one important thing. So that's the reason why uptake is going to be there. Our body understands that some pathogen has entered into our body. So body will try to provide a response in the form of production of antibodies. So here antigens for controlled delivery is possible to the dendritic cells, and the selective activation of cytokine genes in the dendritic cells, and they are useful as therapeutic vaccines, and in the form of cancer, AIDS, hepatitis B vaccines, and HC vaccine, hepatitis C vaccine. So here therapeutic vaccines, you have to understand, most of the vaccines are given as prophylactics, as a means preventive. But if already infection has entered into our body, then also you can provide some relief by giving these particular vaccines. And that is the job of therapy or therapeutic vaccines. That's also another strategy scientists have done. And so you can use them. And here this particular picture shows a confocal microscopy picture in which antigen presenting cell, that is what is known as DC, is nothing but the dendritic cells. Dendritic cells have taken the polymeric materials, and you can see how that means it absorbs all the polymeric material inside, and it shows you glowing color. It indicates that better uptake is there, and that is what normally happens in the body. And this picture shows what is known as a single dose vaccine delivery system. So here single dose delivery system is that for example, if the doctor advises that you have to take 3 doses. So 3 doses means one first dose, maybe 2 months later or 6 months later, you are supposed to go to the doctor, and then 3 times there are cost estimations were there. A child is there, a young working woman is there, and she has to take the child to the doctor. So naturally on the day she has to first take the leave, and she will not be getting salary and transport, etc., and coming back, and after 2 months once again, like that. One vaccination requires 3 doses, 3 times she has to take leave and go. So all the cost is calculated. So today I don't have to earn, because I cannot go to the job on the 2 months later, 6 months later, like that. So if you provide a vaccine which prevents the frequent visit to the hospital, that saves you a lot of time and money. So that is the basis for this kind of trials. The single dose vaccinations and the cost involved, and the companies are putting a lot of money doing this. So different batches of how they are prepared is that in a single dose, different batches are there in which a big particle will be there, small particle, like that. It's a mix. So different batches of microparticles with different antigen release rates, because a pore size may be big, pore size may be less, like that. Sizes, pore sizes, etc. Variety of tricks they try to see that the delivery or the release of the vaccine candidate vaccine antigen is monitored in such a way that first it gives some dose and later some dose and later on some dose, so that 3 times dose is given to the body with one single shot. So that is what the job. And here the polymers like polylactic acid glycolic acid, PLGA, has been used by many scientists. Polylactide, co-glycolate, microparticles are there. Antigens like tetanus toxoid, diphtheria toxoid, hepatitis B antigen, and HIV, means that is human immunodeficiency virus, AIDS virus, GP120, etc. There are so many scientists have worked on this. A lot of research is there, publication. And okay, so now I told you a little single, and now I told you, so for example, okay, single dose. The non-invasive means if you, instead of giving a dose through the body by injection, if you take the oral route, say oral polio vaccine you have had. So nasal vaccines are also there. So these are all considered as a mucosal vaccines. That is through the mucosal route. Because in our body, see, even coronavirus, you know that it enters into our body through this. See, there are waterborne infections. If you take contaminated water, that means they enter through the water that you inhale, or contaminated food. That means actually the route of entry into our body is many microorganisms gain entry to this nasal or the mucosal of the oral route. So naturally, this mucosal route is a good route. If you can prevent the entry of the bacteria or virus at that particular place by defense, our body will be protected. So that is, and moreover, easy, because nature has given the mouth to accept anything from outside. So acceptance or the compliance otherwise is very high in the case of mucosal vaccines, and a lot of research is done on this area. So oral or mucosal vaccine, the basis for understanding is that if you take oral route in the body, it goes like this, and it is a large intestine, rectum, and other area. So here in our body, we are having what are known as, say, Peyer's patches. So this picture is showing the Peyer's patch, and we'll go back here. So within this Peyer's patches, you are having, there are some cells known as the M cells, or otherwise known as microfold cells. And these microfold cells are unlike the other cells which are present in this thing, and they have the ability to accept the big particles. Normally, you know that the molecular weight and transport and absorption, you must have seen for the drug molecules, small molecules only will be taken into the body. But here, antigen means it's a big molecule. So these are all taken into the inside of this body by means of the M cells, and there's the microfold cells which are specifically present, and they accept it by means of phagocytosis, etc. And then these particular vaccine, the antigen is actually taken over by the macrophage, and then some amount of antigen is there to the B cells and T helper cells, and both of them are got activated, and a mesenteric lymph node and thoracic duct, and then ultimately the whole cells, that is the blood, B lymphocytes will be present in the blood, and these cells will go back to the, say, mucous membrane or underlying mucous membrane, and they try to produce the antibodies, and these antibodies are going to be dimerized, and these dimers are known as a secretory antibody, secretory immunoglobulin A. So this cell which is capable of producing antibodies is called the plasma cell, and the B cell is not capable of it produces, but it's not fully mature, and when the fully mature antibody producing cell is there, called it is plasma cell. So naturally plasma cell. So this is a simple oral route you can take. Antigen is uptaken by the M cells, and then they are processed, and the B lymphocytes are activated, and the T cells are also activated, and they provide mature plasma cells, and these plasma cells will be transported through the blood to the mucous membrane, that is underlying mucous membrane. There they try to produce the antibody. Antibody will diffuse from inside to the outside, and the antibody will diffuse from inside to the outside, and it will cover this. So naturally, when an X organism is coming through the mouth here, the antibodies will try to, say, protect the attack from the infectious organism. So that is what the basis for the mucosal vaccine. And here you see it's an anatomical picture, histological picture of the what's it called, Peyer's patch here. And here you see this area is called the dome area, and these are the villi, and you see that in these dome areas you will be seeing the M cells. Here the M cells will be there, and whatever the picture I told you is actually the microscopic slides that you are seeing. So here Peyer's patches are present in gastrointestinal tract, but it in the colon region more and the rectal region. Jejunum and duodenum, they are somewhat less. Patch contains, one is dome area, follicle associated epithelium, follicles of B lymphocytes are present, and Peyer's patch is covered with follicle associated epithelium. It is in short known as FAE, consisting of M cells. Usually the number is colon region 15 Peyer's patches per square centimeter, 25 per square centimeter in rectum area, and the size of Peyer's patch is 1.5 to 3 millimeters diameter, and sometimes small may be there. 2 to 3 centimeter millimeter diameter, 7 to 9 means big aggregates will be there in the form of this thing. And here the epithelium is enriched with lot of antigen sampling cells known as microfold cells, which I told you. Goblet cells are not, means mucus secreting cells are not there usually. M cells lack the brush border also and transport macromolecules. So this is the nature you see. I told you they don't have the brush border, and this type of microvilli are not there for this particular cell. So here Peyer's patch, and rabbits contain 40% of follicle associated cells are M cells. In rodents it is human beings also only 10% are, and Peyer's patch consists of cells necessary to induce and regulate the immune responses. Here the both type of responses, B cell follicles and then T cells are also going to be activated, and things like that, and the rest of the story is more or less same. And here what exactly is important is that the microfold cells and the antigen, you can create some environment where the uptake is going to be there. That is M cell targeting is also there, we will talk later. So this type of things are going to be there. And here in this picture, the same one what we have just now showed, this is M cell and antigen is there. Antigen is taken by the M cell here, and dendritic cell gets activated, and here you are seeing the CD4 T cell is there and the B cell is there, and both of them are going to be activated. Here the lymph nodes and then mesenteric lymph nodes are there, and thoracic duct, whatever the picture just now I have shown, that this is inducing antigen. And the same is transported here through the blood and produce the interleukin. Plasma cell is there, and antibodies are produced in the form of dimer. This is one molecule, this is another molecule, it's a dimer. So that is same picture just now shown here is once again shown in the color. So here just now I told, so mucosal vaccines are there. And what is the rationality and basis for the some mucosal vaccines? The rationality and basis for the mucosal vaccine is that underlying one phenomenon is the common mucosal immunity is persisting. So common mucosal immunity is now divided into NALT, GALT, LIF, BALT, etc. are included. MALT means mucosal associated lymphoid tissue. BALT means bronchus associated, large intestinal follicles, and GALT means gut associated lymphoid tissue. NALT means nasal associated. So like that, in all these mucosal area, underlying mucosal area, this type of cell mechanism is possible, and it is common also. So that's the reason why this is called common mucosal. And the antigens, if you are given nasal route, they will get simulated, they will go through the oral route and the total GI tract. Or if you are given oral route, and the antibody producing cells can also come to the nasal route. So this is what exactly the meaning. And the antibodies produced are the dimers, that is two molecules are together, it is called as dimer. And then secretory, they are secreted, so therefore they are called as secretory antibodies. And this is the first line of defense, and it will try to take care about the entry of the pathogen in the body, so through the nasal route. So here this is pictorially shown. Nasal vaccination is easy, oral vaccination easy, rectal vaccination also for children. You also, as young babies, you can insert something, the rectal suppositories or something like that. You can, rectal vaccinations are also there. So associated lymphatic tissue, nasal associated, gut associated lymphatic tissue, and large intestinal follicles are, these are all going to be there common. And then immunoglobulins are there in the upper respiratory tract, immunoglobulins are there in gastrointestinal tract, immunoglobulins in the exocrine glands like mammary, salivary, lacrimal glands, and in the urinary vessels also they are going to be present. So here, wherever the mucus lining is there, the antibody producing cells spread and they try to produce cover of the antibody, and that actually prevents the entry of the bacteria or the viruses. And you can, as a matter of fact, improve what is known as antigens specifically to the M cells, and that concept is called as M cell targeting. So here, how the M cell targeting is possible means, so normally M cell has the ability to take the antigen, but you can specifically provide some ligands so that the M cell targeting is enhanced, the rate at which. So here I show you, because I'm not going to discuss, the time is very little. MABs, that is specific MABs are, that is monoclonal antibodies are there for M cell surfaces. Example, you can take monoclonal antibody 5B11. This reacts with the brush border of the enterocytes. And similarly, lectin-based Ulex europois 1 agglutinin, UAE agglutinin is a M cell lectin, and it can also be used. And CLI-related Lewis antigen was also tried. So these three categories are going to help in what is known as the M cell targeting. M cell targeting means there are so many cells in the body, but our antigen will only go to the M cell, and the uptake is possible from the M cell. So this is what exactly basic targeting. So here, next, mucosal immunization. Just now I told you can put the nasal route and then oral route, etc. And antibodies are produced very good. Almost 70% of the antibodies in our body, if you take and measure, that is almost 70% of the antibodies are there under the mucous membranes. So this is an important thing, and you can use this particular concept for the develop of this thing. And their advantages, if you see, there usually preferred route of a non-invasive, cheap, and high compliance means a child will be very easy to take nasal route or the oral route, or even rectal also if he is not cooperating, but there is no pain. And to improve the character of these nasal toxins, etc., nasal vaccines, you can use some say potentiations or the adjuvants. Let us say cholera toxin can be added. Similarly, salmonella, live vector can be used, or some other microparticles can also be used to improve the adjuvanticity. And examples of mucosal vaccines include salmonella typhi TY21A, cholera vaccines, cholera virus D103, HGR. This is attenuated, and you can give in the form of oral polio vaccine, just like salmonella. Salmonella means for typhoid vaccine, you can give. Similarly, COVID, CVD, not COVID. CVD is nothing but cholera vaccine. Also, oral route you can give children. And oral polio vaccine is so popular that you know it. Helicobacter pylori, urease vaccines are also there, and some research is also going. That means mucosal vaccines have become very popular and easy to administer, easy to take, compliance, and the good immunity is there, and research is going. And if you take mucosal this thing, these factors which influence the microparticle, because I just now told you that microparticles are one systems which you can use them. And here, when you are designing microparticle size delivery systems, particle size, polymeric composition, polylactic acid, glycolic acid, 50-50 or 60-40 or 75-25, like that, particle surface charge, whether it's a positive charge, how much of positive or negative, dose of the particles, how much you are given, administration vehicle, and animals you have used for studies, and then age of the animals, and whether the food is taken or no food is taken. Variety of factors are going to influence in the analysis and studies. One has to be careful about that. And sometimes what happens is that if you are really interested in putting your antigen across the say biological membranes, and as such the membranes are not permissible. By nature, our membrane doesn't permit, and biological membranes only accept small molecules around 400, 300, 500 molecular weight, and some lipophilicity is there. That means we measure in terms of log p value and not small molecules. They are not usually there for the big molecules, macromolecule. So then to improve through the oral route, the molecules or the macromolecules, the scientists have done some exercises by understanding the nature of the membranes, and they have tried that using some permeation enhancers, or otherwise known as the absorption enhancers, they try to put some influence over the biological membranes, like for example transdermal here the what's it called, the transcellular and paracellular. So these two types of things are going to be disturbed, and how they could do this, that they try to disturb the structure of the existing biological membranes, and then they try to push the materials across the biological membranes, and they will be able to provide the absorption or penetration. And there are variety of mechanisms have been suggested, and then the advantages are there, sometimes toxicities are also there, and I'm not going to discuss much about this because it is usually you might have studied sometime. So microspheres, if you put the antigen, and these microspheres release the antigen very slowly, and this is actually acting as depot. Slowly it releases. Actually an antigen must be very effective, means it should release the antigen very slowly, and the microparticulate systems will provide this particular one. And the sizes may be say 10 microns or sometimes less, and you can use. So the important point in the uptake is that macrophages are the say antigen presenting cells have to phagocytose it first, and then only they try to provide the release of the antigen, and the released antigen is exposed on the surfaces in the form of MSC class 1, MSC class 2, and then the T cell receptors are going to be there, and then they are getting activated, and then the two wings, that is humoral immunity and the cell mediated immunity, are activated, and then the antibodies are going to be released. So here the antigen release, so gradual release is possible, and you can also in the form of burst or continuous or pulse like things, you can control this variety. And there are variety of antigens have been tried by the scientists: Staphylococcus enterotoxin, diphtheria toxide, and hepatitis B surface, and tetanus toxide, bovine serum albumin, PLGA, PLA, PG. These are all polymers. PLGA means lactic acid, glycolic acid, only lactic acid, glycolic acid, polymer of glycol. So the methods like solvent evaporation, WOW emulsification, phase separations, and suspension, emulsions, and adsorptions. Different scientists have done research work on this area. And when you prepare the microparticles, pulsatile, at one shot, lot of antigen is released, and some burst release, and then followed by pulse, or sometimes gradual release. So these are all the same type of knowledge that you do for the simple drug molecules, drug delivery, the same thing. And here you can say analyze these particles like chemical analysis. What is the chemistry, degradation kinetics, density, isoelectric point, because the macromolecules are having particularly proteins. If you take isoelectric point is important one, and the isoelectric point is the point where the pH, the macromolecule is unable to move, but that means it loses a charge, and they try to precipitate, and the solubility is reduced, and when the solubility is reduced and the release is not possible. So there are so many important aspects pertaining to say isoelectric point, and how much you can load here, and the release rate of release, etc. And these are all the things which are there. And the two important methods are there for preparation of the microparticle. And here one is using organic solvents, but nowadays organic solvents are not advised, but still there is a method: polymer and dichloromethane, and aqueous consisting of antigen. Example is the bovine serum albumin, W1, and when you mix them together, it results in emulsion, that is W1 water inner phase and outer oil phase, and water plus polyvinyl alcohol, it is once again W2 phase, 1% homogenization, you will get WOW, one internal, outside, external water, internal oil. And the solvent evaporation, DCM, dichloromethane, which we have used evaporation, you will prepare nanoparticles or the microparticles, as the case may be. If the rate of shear and mixing, etc. will tell, and then you wash, lyophilization, so that all the dichloromethane is removed, but still there will be some residues. And the Food and Drug Administration usually do not admit, and you have to prove that your residual solvents are very, very, very negligible probably. So then only, and then alternative scientists have developed one more protocol where you can prepare without any organic solvents, microparticles or microspheres. And here aqueous solutions. And for example, if your protein is an antigen and it is dispersed or mixed with dexhema, that is dexhema is hydroxyethyl methacrylate. It is a dextran linked. So this protein is there, and the polyethylene glycol, if you mix them together, these two are normally separated, and when you mix it, churn it, stirring, you will get some globules like this, and the protein is dispersed. Now it is in the form of water and water emulsion, water in water emulsions. Polymerization, you will get microsphere. suspension and washing, freeze drying, you will get the particles. So water, only water phase, no organic phase. So this type of what is called dexhema, that is dextran related and hydroxyethyl methacrylate particles are there. So now if you have prepared such a type of say particulate system, and you can just see here in this picture, what you are seeing is that an antigen is concentration, that is in the body it is related. 3 injections are this is related to the single shot only, I'm telling you, traditional method. So 3 injections are to be given in this particulate. First dose antigen is released, and after maybe 3 weeks or 4 next dose, and next time 3 times 3 injections are given. And here if you see, in this case, because of this 3 different categories of the particulate systems are pooled in this particular injection, single injection. First dose is there, and second, third. So whatever first you want, you got here. Second, you got here. Third, you got. So it means that a single dose consisting of different particle sizes prepared with polymers with different release kinetics can be pooled and prepared in the form of one single shot, and it saves you going to the hospital 3 times, 3 days leaves and the transport, and so many things will be avoided. So this is all behind this. So now you can, scientists are doing like this. And how you can control this is actually what is known as these polymers. And then the important point is that degree of substitution, the dexhema degree of substitution is important. And initial water content and the in vitro release can be controlled for this particular antigen. And here, so here the pore size is somewhat less, very fast release, pore size somewhat very less, then release is reduced. That means if you move from here, the antigens are not going to come out from the polymeric matrix, and the pore sizes are very small. So that means you can avoid the release or retard the release. That's the what exactly means. So how you can do that? By the substitution, that is in the polymer, whatever the things that you choose, the degree, extent of degree substitution or otherwise reaction, you can control such that different pore sizes may be there and you can use it. And here, important considerations in the single shot vaccine development. One point that you have to see is that biodegradability of the, but whatever that you have taken, injection, the polymer is degradable. That means it should not be there in our body after some time. That is called biodegradability. And encapsulation, how much antigen can be put, that is another thing. Particle size, which means very small, big, medium size. And then preservation of bioactivity. So in the process of preparation of the antigen in the form of microparticles, the antigen character has not lost. That means the bioactivity is not lost. That also you have to ensure. Scalable production process. In the laboratory, you can do successfully. But if you want to prepare in the industry, let us say 1,000 liter batch or 2,000 liter batch or 10,000 liter batch. So then the scalability problems will be there. And effects of combination with other adjuvants, and whether they can be given by means of different routes of administration also you can check. So this is all regarding single shot vaccines. I also told what is known as mucosal vaccines. Now I tell you something about what is known as DNA vaccines. Basically, what is an antigen? If you look into the structure of an antigen is a macromolecule having usually about above 5,000 molecular weight. But basically, to be good antigen, at least it should have 10,000 molecular weight and above. So the molecular weight is more, means the complexity of the molecule is going to be more. And when the complexity of the structure is there, usually our body considers it as a foreign material, and the body will respond in a different way by producing antibodies against them. So that is what exactly the reason for the antigen. So this antigen structure is to be protected in the body. And the antigen structure is the one which is recognized by the body. And if the antigen structure is lost, our body cannot produce antibodies against the same. So that means, for example, if you take a protein, nature protein and denatured protein. Natural protein is the 3D conformation is intact, and the natural protein is heated, let us say, put in some solution or some acid, etc. The 3D structure is lost, and therefore you may not get it. So now, so this is protein structure. You can give protein in the form of injection by putting in a microparticle or nanoparticle or some other ways. Or instead of a protein, you can give a gene. So normally proteins are produced in our body by the genetic mechanism. In our body, chromosomes are there. Similarly, that foreign protein, for example, having genesis from the virus are having genesis from the bacteria. That protein will have a particular gene, and that gene also can be taken instead of a protein, and that gene can be put in a vector, and that vector can be administered. When the vector enters into our body, that DNA is transcribed in the nucleus to get mRNA, and an mRNA is translated to get you protein, and that protein is active protein, and active protein is present in my body, which is foreign in nature. So naturally our body reacts in a different way, and that will try to produce the antibodies. So that is the logic behind the DNA vaccines. So here recombinant DNA strategies, recombinant DNA technology is taken into account. For example, here immunological protein of interest, for example, any protein, let us say hepatitis B, let's say surface antigen, or sometimes nowadays spike protein, coronavax spike protein, we also discuss a little later. This protein is there, and that protein is produced by this particular gene. This is called the corresponding gene, and this gene is actually put in the gene, means the DNA sequence, and the plasmid is a small, say, extra chromosomal DNA molecule. Within that, this particular gene is inserted. You are getting the plasmid with this particular gene. So when it is transferred into the cell, it is called transformation. The cell acquires this particular one, and the host cell is there. And for example, here, like a virus, like vaccinia virus, etc., is also there. And this viral vaccine, viral DNA is entering here. DNA is there. These two. What is known as recombination takes place, and this portion consists of this thing, and then this cell is getting destroyed, and the virus, the whatever that particular thing with you were here, is now entered here. This recombinant virus is capable of infecting these cells, and this also antigen is transferred to these cells. That is what exactly the basis here. So now once the antigen is transferred, you can use it. So here I am going to discuss a little bit about a virus vector known as adenovirus. Adenovirus means adenoviruses are nothing but the DNA viruses. So adenoviral vector is used, and in the corona vaccine, DNA vaccines, adenoviral vectors are used. And here the adenovirus is modified DNA. This is the actual piece of DNA which is not important in the adenoviral DNA. This is removed. So in this particular case, the disease, the gene responsible for the cytokine or the whatever the antigen containing gene is inserted here, and this is once again inserted back in the virus, the adenovirus. The earlier portion is removed, and it is inserted, they say, and it is once again made. And now this particular virus is capable of infecting this, and then you see that this particular gene is producing mRNA copies, and then a lot of antibodies are going to be produced. So this is how you can use adenoviral vectors. And some scientists have also tried say bacterial cells also. Bacteria also infect us. So through the bacteria also you can put your genes and into our body. That's also there. The BCG was also tried by some scientists. So one more concept is that the DNA, just now what I told you, is viral vectors. That means a virus, vaccinia virus you can use, or adenovirus you can use, or some other viruses can be used. But the only thing is non-replicating, non-infective type of viruses you have to select. And within that virus you put, and that virus will act as a carrier for the delivery of the antigen containing gene. So here, sometimes scientists have also tried what are known as DNA vaccines only, but naked. Naked means they don't have any kind of say viruses structure or something. Simple DNA molecules there. And the advantage of this type of is that they can be given to the oral, dermal route, intramuscular, nasal, or mucosal route. No cold chain. There is no, because it is only simple chemical as a DNA molecule. So you can put it room temperature. So plasmids containing genes encoding proteins having antigens are prepared, injected, and they are simply injected, which contains the genetic information. And they try to provoke the antibodies. So rabies vaccine was tried, rotavirus, hepatitis, herpes genitalis, and then influenza, simian, AIDS, etc. There are so many vaccines. They are called as a DNA vaccine, but more specific naked DNA vaccines. But the problem is that integration of this nucleic acid resulting in tumorigenesis and immunological tolerance. So this is all one important. That means what exactly is that this DNA is to express. Express means that DNA is to be translated. So translated means DNA is made in the form of mRNA. mRNA is translated to the say protein. by means of ribosomes. So these are all the transcription and translation. These two stages are there. But the DNA, since it is a foreign DNA in our chromosome, it can go and integrate any place. We don't know. That is what exactly the problem with this type of vaccines. And you can also put what are known as the dermal vaccines. That means through the skin. So far adenovirus vectors I told you, and then the dermal vaccines. That means through the skin you can also put this thing. So normally if you say skin consists of what exactly, the first portion, stratum corneum. From here to here you will be having basal lamina, and then from here to subcutaneous tissue. So if you take normal injection and put a needle like this, and if it is up to this portion, let us say from here to here, it's a lot of pain, because this redness in way increases the pain. And by some way, the needle is only up to this, the pain is not there. So that is what exactly the thing here. Here in the dermis region and then epidermis region. So there are some vaccines have been developed, some machines are developed wherein this machine will try to give you vaccine through the dermis only. So here it is epidermis only. This is called EasyVax, and the dermal vaccines are Dermavax. These are up to this only. That means the penetration of the particular needle is only to the superficial layers. So once it is not going deep inside, the pain is not there, and it is going to be easy. So this type of concept is also there in preparation. And here there is a small machine. I hope you are all able to see this. This machine is there, and the delivery process, Dermavax delivery systems, and the vaccine is inserted into the skin using a small hypodermic needle, and then insert needle electrode. That is, there is one electrode is also there into the skin, 2 millimeters, only 2 millimeters depth, and some voltage is applied, and you will be able to see that the penetration of the drug molecule inside. So you can manipulate the system here from outside, and you can control through the computer, and compliance is high. And so this is some machine developed for the Dermavax. That means the Dermavax helps the delivery of the vaccine through the skin to a very not very deep but superficial layers. And similarly, EasyVax is also another delivery system, and it is something like a gun, and you can quick fire this and all. And here, best suited for mass scale immunization, prophylactic purposes. Thousands and thousands of people, if you are interested to vaccinate, this type of delivery systems are very, very important. And here, this EasyVax only concentrates on the epidermis layer of the skin. Just now I have shown you. And it consists of two component system containing a mobile electric field generator. That means this you can carry this in the handpiece, and disposable microneedle array. So microneedles are very, very small materials. They are needle like, they can coat them. So an antigen can be coated. That's microneedle technology itself is one big lecture. So you can use them, and these microneedle arrays, you can put them, and they will be only penetrating into the skin. That's all. So the DNA can be coated, or some other protein may be coated, and etc. You can insert them this microneedle array, and you will not see any kind of pain, etc. And you can deliver very quickly to this. The machine that you can use is the EasyVax delivery machine. So like these machines you can use, and mass immunizations can be done. Now, after I hope I have given some idea regarding what is known as the vaccine, current and traditional vaccines. And I also told a little bit about historical aspects, how they have done, and the challenges also I told you. And to overcome the challenges, the WHO and other companies are encouraging some novel strategies to prepare vaccine delivery systems where more immunity, safety, and other route of entries like nasal, mucosal, and then rectal also, and variety of things have been there. And after listening to the to get better efficacy, transdermals are also tried, like transferosomes, and then proteosomes, virosomes, and aquasomes, and etc. And there are many other such delivery systems have been developed by scientists. And now I'm going to give maybe 10 minutes also. I don't know whether Dr. Gopinath, whether I have some time. Dr. Gopinath. Okay, I'll continue. Maybe in 5 minutes I will close this. So
okay,
now we're all in an era, unprecedented era of corona vaccine, the corona period. It's a COVID-19. And this picture shows you the causative organism, that is a coronavirus. So now the whole picture, these are called the spike proteins. These are basically the things which are now more interesting. And inside, if you see, it is a RNA virus. Some nuclear protein is there, RNA is there, and it's an envelope. And heme agglutinin dimer is here, and a spike glycoprotein here. So this is a structure. Basically, it's an RNA virus and it's an envelope virus. RNA, some nuclear proteins are also there. So this is the structure. And now you know that this particular virus is emerged from Wuhan in China in last year 2019, maybe in the November or December, and it is spread like anything throughout world till now. Due to this particular disease, it's called as COVID disease, COVID-19. Almost 2.7 million people have died. It's a big in the history of say mankind. Okay, in swine flu, Spanish flu was there in 2000, in the 1900, early 1900s, etc. But in this particular current scenario, because of the advances in the sciences and life sciences, medical sciences, etc., more than 2.5 million or 2.6 million till now are not dead by this vaccine, by this particular virus. It's a very dreadful disease. And now we'll talk a little bit about this. This virus is known as SARS, because this SARS is nothing but it is based on severe acute respiratory syndrome, SARS. Earlier, it was an outbreak in 2003, and MERS means Middle East respiratory syndrome. It's another outbreak in 2012. They have killed some thousands of people, not as many as this. So this virus is actually related to these earlier viruses. SARS-CoV, this is related, and it is now what's it called, is belonging to corona viridae family. This family is based on what is known as RNA based viruses, and this is till now 2.7 million people have done, and it is emerged in Wuhan city 2019, and it is called coronavirus disease. And actually, as a matter of fact, if you see, unfortunately, there are no good treatments because it is emerged very quickly, and some treatment protocols involved the repurposing of the drugs. That means the drugs which were developed for some other purposes, lopinavir and ritonavir, which were developed for the HIV and were tried, and they actually involve a protease known as a chymotrypsin, like protease inhibitors were tried, and along with that remdesivir, dexamethasone, and hydroxychloroquine, etc., were developed, and not developed, they are existing. They were tried to reduce the mortality and hospitalization period, etc. So, but still, it's not 100% cure. There is no effective drug as on today, and but you can reduce the mortality. That is the advantage. So here, then, what is the thing left against so many deaths? And alternatively, what you can think is known as a vaccination. Vaccination is one thing which you can prevent. So, so vaccination strategies have been considered for this RNA-based virus. And I don't know whether we'll be able to get this video. I don't know how. No, I think it is, sorry. So this virus, this coronavirus, when the antibodies are produced, they try to actually interact with these antibodies, will try to interact with this. So this is what exactly the virus is going to go over this. And different vaccines have been developed throughout the world. There are so many say companies, research scientists concentrated much in this particular area. And I'm going to tell you a little bit in the next 5 minutes or so. So in our country, if you see COVID-19 vaccines, one vaccine is known as a Covaxin, Bharat Biotech company in Hyderabad based. And actually they have developed this vaccine, and it is inactivated indigenous viral vaccine. In our country, it is developed, and it is approved for emergency use, currently used in older people above 60 years. And what exactly it consisted is inactivated SARS-CoV-2 virus, SARS-CoV-2 virus, and this specific number of the strain is NIV, National Institute of Virology 2020 770. So this is a code or the strain number, which is disabled for replication, means it cannot multiply. It cannot multiply. That is what exactly the thing. And the proteins are intact. That means this is a total virus. No proteins, not spike protein. Just now I have shown a spike protein. It's not like that. And the immunity is vaccine produces antibodies against many regions of complete virus. Intact virus contains almost 29 different types of things. The immunity produced by this vaccine is more comprehensive and closer to the natural immunity. And the components, if you see, it consists of aluminum hydroxide gel, imidazopinone. This is an adjuvant, and this adjuvant is the TLR 7/8 agonist. This imidazopinone is it can bind to the TLR, that is toll-like receptors on the cells. So here 2-phenoxyethanol is another component there. Phosphate buffered saline is the biotech have actually developed, and this aluminum hydroxide gel is used as adjuvant, and which stimulates immunity. The Th1 immunity means antibodies, humoral immunity, which produces sometimes this is also there produced by the RNA vaccine. Bacteria, this Bharat Biotech actually have done clinical studies, phase 1, phase 2, phase 3, etc. are there. Around 25,000 people have been included in this, and 2 doses are advised. It can protect the people against say possible mutations or the variations in this. So this type of vaccine is also there. And then Covishield is also another vaccine which is developed or manufactured, as a matter of fact, not developed, by the Serum Institute in Pune, not Serum Institute, Pune. So here they have taken the technology from what is known as Oxford University and AstraZeneca. This AstraZeneca is a company, university department. They have collaborated for development of this particular vaccine, Covishield. And what exactly they have done is they have taken the vector, and which vector? Adenovirus vector. Just now I was talking about the adenoviruses also. They have used the same, containing a gene for spike protein, and used in this vaccine. And this they have given some code number AZD1222. So this is a code, and usually what the adenoviral vector is that this virus is weak and they cannot replicate, and the virus is actually obtained from the chimpanzee. So the chimpanzee adenovirus is taken, and they carry this particular gene, and therefore it is ineffective in the human beings and used in the individuals more than 60 years. And this protection usually starts 15 days after the second dose intramuscularly it is given. And the composition include histidine is an amino acid, and the histidine hydrochloride monohydrate. It's magnesium chloride is there, and the polysorbate, AT, ethanol, sucrose, sodium chloride, disodium, EDTA, otherwise water for injection. You can store the same in 2 to 8 degrees centigrade. 2 doses are required. And this produces antibodies against the spike proteins, S proteins only. The DNA needs to enter into the cell. The mRNA will be synthesized, which comes into the cytoplasm and gets translated by the ribosome. The method how it works. The immunity produced is not exactly as produced by the Covaxin. Whatever the Covaxin production immunity, this is slightly different, not the same. And other companies in the United States of America, they have produced what is known as Pfizer company, BioNTech. So these two companies have come out with one candidate vaccine, that is BNT162b2, and this actually is based on mRNA. So the earlier one is adenoviral vector they have used, and now they are using mRNA, messenger RNA. This mRNA is synthesized in the chemistry laboratories, and this mRNA is actually mixed with some lipids. So a complex, that is mRNA lipid complexes are prepared, and they are injected as a matter. That is what exactly. mRNA based combination of 4 different lipids, and lipids are not clearly known. And sucrose is used as a cryoprotectant against the temperature, and it acts also stabilizer. More than 90% efficiency. 2 doses are required with a gap of 4 weeks, and around 46,000 people have been there in the clinical trials. And very cold storage. The important problem in this mRNA is that it is very, very unstable. Almost 94 degrees Fahrenheit, means let us say minus 60 degrees centigrade is Fahrenheit. So if you are interested to do this, transportation is big issue, and dry ice can be used for the transport in the containers for shipping purposes. High level of protection is there against COVID-19. Protection starts after 7 days of the second shot. And in the United States of America, there is one more vaccine known as the Moderna vaccine. this Moderna vaccine is also actually mRNA vaccine. When the mRNA was mRNA virus genome was published by the scientists, that is first they have in the Wuhan they are isolated, and actually this virus was considered as propagated from the bats, means in Telugu it's called gabilam. So from the bats it is there, and immunity is immediately after the publication of the SARS-CoV-2 virus RNA genome structure, and they have taken the this mRNA gene pertaining to what is known as spike protein, pertaining to the spike protein that they have taken. This is a candidate is mRNA 2L73, and then once again lipids were mixed and they made some complexation, and the mRNA was basically a synthesis reaction. And the advantage is that once you have injected, mRNA is immediately translated by the ribosomes present in our body. So that means immediately protein is produced. That is a spike protein is produced. And the spike protein is produced means the spike protein actually triggers the immunological reaction. So that is what exactly. And the advantage of this technology is the scale up is possible, mass scale, you can produce any number. In the clinical trials, they have used almost 30,000 volunteers and 94.5% efficacy. And 14 days after the second shot, you are going to get the what's it called? The immunity starts, and it is basically injected. That's what exactly I told. And the storage is also very low temperature, minus 60 or 70 you have to store. And the problem of mRNA is instability, and degradation may be there in 5 days. And the dry ice, just now I told you, first dose after 3-4 weeks gap, second dose is to be. And this mRNA cannot be transmitted from one individual, from me to my son. It's not possible. Next generation. After injection, 24 to 48 hours, the viral protein starts producing. So this is regarding the mRNA. That is Moderna vaccine, and then BioNTech, that is Pfizer vaccine. These are now taken or given in America. And now you are having one more vaccine that is under the clinical trials produced by Johnson & Johnson company. And this Johnson & Johnson company produces something like, say, Covishield type vaccine. That is adenovirus vector they have used. This adenovirus vector is called 26. And spike protein genes are present. Protection starts 14 days afterwards, and almost 70% protection. Vaccine and storage at 2 to 8 degrees centigrade. So this is regarding Johnson & Johnson company. And a Chinese, Russian vaccine is also known as the Sputnik V. It is called as the Sputnik V vaccine. And Gamaleya Research Institute of Epidemiology and Microbiology. Adenovirus viral vector vaccines were there. And here, this SARS-CoV gene, once again, here it is there. And what they done is 2 different type of adenoviruses were used. Say one is AD26 and AD5. In the first dose, AD26 vaccination is given, and the second dose, AD5. So this is their strategy. And the only thing is they are also viruses, non-replicating viruses, and within the DNA, only they are present, but 2 different viruses they have taken. That is their different strategy to get to some, what's it called, patenting or whatever that is there that they have taken. And that is also found to be efficacious, and it's also giving response in Europe. That is European Medicines Agency has approved this Sputnik vaccine. And in the Chinese government has also, this Chinese company known as Sinovac is a biotech company, has actually produced this inactivated vaccine. Inactivated COVID-19 vaccine was there. And this virus is SARS-CoV-2. This is a virus. And how this virus actually is cultivated means in varocell. This monkey kidney cells, this varocell grown, that is in the animal cell culture laboratories. These viruses were cultivated, harvested, and then they are inactivated because the cells were killed. And then aluminum hydroxide was used as adjuvant, and other buffering agents like sodium dihydrogen phosphate, monohydrogen phosphate, sodium chloride, etc. They are simple preparation and intramuscular injection, 2 to 8 degrees and 83% of individuals in clinical studies, they say in Turkey, that is where about 10,000 people were there in the clinical trials recruited. So this is Sinovac vaccine. And apart from these vaccines, one more vaccine known as the Bharat Biotech company is actually contemplating and doing some research with this candidate, BBV154. It is a novel adenovirus vector vaccine, but it is meant for intranasal, that is through the nasal route. Because instead of giving injection means pain, some fever type thing, inflammation, and variety of such complications are there, and I'm not going to discuss about this thing. Intranasal vaccine, they are also trying. The advantage is that it stimulates both antibodies, humoral antibodies, and neutral mucosal antibodies, T cell responses. It is considered very good, and immune responses are found very good. And future, I think, but not, they have not released right now to the public. Maybe in future, intranasal vaccination may be popular, and you can take the same. So this is actually the end of my lecture. And in this particular lecture, what all I have told is what is basic vaccine, and a little brief introduction, and some historical account, and the challenges of the existing vaccines, and how you can overcome the challenges by designing and using different delivery systems known as vaccine delivery systems. And the current research is now, a lot of research is concentrated. Normally a vaccine research program takes 4 years to 6 years. Earlier 10-20 years are there, but it has been reduced considerably to 4 to 6 years. And specifically this corona vaccine is finished almost in 6 months to 1 year. But still, I tell you, corona vaccine has to solve so many questions. They are not answering, because it is for emergency use. How long the protection is given? For example, now I have taken the vaccine. Whether it works after 1 year, not sure. Whether it works after 2 years, not sure. So like there are so many stability issues and immune power issues are still pending. But emergency use people are taking, and government is encouraging, doctors are giving. So now with this particular thing, and I'm thankful to all of you for patient listening to me. And I also thank Professor Gopinath for having given me an opportunity to participate in this faculty development program. And I really enjoyed doing this job. And I thank you one and all for your kind patience and listening to my lecture. Thank you one and all.
Sir, thank you very much, sir. And excellent talk, especially present situation, COVID-19, the pandemic effect. People are afraid in that one, either regarding the vaccination taking or not to take or take. And no, you came to the very from basic aspects of the vaccines, delivery types of vaccines. Excellent session, sir. Even end of the session today, most of the faculty members are present here. And very much thankful, sir. Any questions, please? Topic is open for discussion. My co-faculty members. I told you previously the legends in the pharmacy. Sir also another legend in pharmacy. Sir, go back to the first slide, sir, please. First slide.
This one?
Okay, sir. Okay, leave it, sir. Any questions, please? You're given unmute option, or maybe Madam.
Sir.
Chinna Devi, madam. Any questions, madam?
Sir, good evening, sir. Thank you very much, sir, for this topic, especially.
Please, madam.
Sir, thank you very much for this topic, especially, and thankful to you. And there is no question, sir, but I for convey my thanks for that only I unmuted my mobile.
Introduce yourself, madam. Introduce yourself.
Nice of you.
Myself, Chinna Devi, sir.
Yeah.
Working in Palamur University, Mehboob Nagar.
Yeah. I think we have met somewhere. I don't remember exactly where we have met, but I think we have met once. And I feel like remembering your name.
Okay, sir. Chinna Devi, sir.
Yeah. Because I heard your name, some context, I don't know where exactly, but I have seen it appears I've heard about your name. It's wonderful.
Sir, sir, one question will be there, sir. How isoelectric point parameters will affect, and chat box. How isoelectric point parameters will affect the therapeutic action of the antigen loaded microspheres.
Dr. Gopinath, isoelectric point, once again, will you please once again?
How isoelectric point parameters will affect therapeutic action of antigen-loaded microspheres.
Antigen-loaded?
Microspheres.
Microsphere. Actually, because the isoelectric point is a character of a molecule. So particularly proteins, that means when they have this character, if you are preparing the preparation, by any reason, if the pH is going to be different. When you are preparing the microspheres and handling, because the solution, but because here, whatever the protein solution which you have taken, when you have inserted into the aqueous phase, if any change in the pH, you have to measure it, whether the same pH is maintained or slight changes in the difference in the pH. So automatically there is going to be precipitations of the this thing. That is the reason why you must be very careful about the say preparation of antigen in the microspheres. For normal simple drugs, it's not a big issue. We have a lot of research published with normal drugs putting in the microspheres, and it's not going to make any problem. But it's the macromolecule, the three-dimensional network, a 3D structure of the particular molecule is prone for this thing, and you must be very careful. The meaning is that it's not that always it is going to be affected. It's not like that. The purpose is not that. You must see that there is a possibility for the loss of biological activity of the protein molecule, because the pH is important. And if you are ignorant and if you are not careful about the pH variation during your processing, then there is a likelihood that the precipitations are going to be there and the charge nucleations are going to be there. So therefore, you must be very careful about it. That's what exactly the meaning. It's not that when you have used the microsphere preparation, it's not compulsory. And so sometimes it may happen. We don't know exactly what is the protein and what is the nature of the protein and what temperature, what pH is more sensitive to that. But insulin is having one pH and some other protein is some other pH isoelectric point, and we don't know exactly. That's what we have to see. I hope I think this should clarify. Dr. Gopinath, I am unable to listen to me. You listen to you. Which is please unmute.
Okay, sir. Sir, another question.
Yes.
Sir, my parents feeling tension to take coronavirus vaccination. Please guide regarding.
Actually, how old are your parents?
Please write in chat box, or maybe you can raise hand raise, please.
How many years
What
old?
is the age of the parents?
Huh? What is the age?
They're not mentioned, sir. I'm asking. 65 years, sir. 65 years.
Then they are taking
65.
70, 75 also people are taking. Only thing is the advised thing is right now is that if somebody is allergic and there is a doubt allergic to what, so some doctors say that the components which are present in the vaccine. So vaccine contains some, for example, aluminum hydroxide or some other such component is there. If they are having such kind of history, better not take allergic. And otherwise, blood pressure, hypertension, oblique hypertension, or even diabetes, the doctors are advising you can take. There should not be any big problem. Some even heart problems are. But some selective cases you have to see. That's also they are telling. In general, without any such kind of comorbidities, they are called as comorbidities. They are advising to take. And there is also some section of people are not taking it all. It's because if they are at home and not going outside. So let us wait and see. That's also some people are doing that. But 65, 70, 75 also they are taking.
Okay. Any questions, please? Sir, excellent talk. Sir, what are the, sir, my...
Unmute, please. Sarita, madam.
Sir, another question, sir. Sir, another question.
Yes.
From the participants.
Unmute,
Another
please.
question.
Gopinath, sir.
Sir, is it too safe to take vaccine for? Okay, listen. Sir, is it safe to take vaccine for thyroid
Loud.
patients?
Bit loud. I am unable to listen to you. You please.
Is it safe to take vaccine for thyroid patients?
Safe for?
Vaccine is safe or not for thyroid? Thyroid patients, sir.
Thyroid patients.
Thyroid, yes, thyroid patient. Yes,
Thyroid
patient.
Regarding COVID vaccine, you mean to say which one? Which vaccine?
Yes, sir. Yes, sir.
COVID vaccine.
It is the COVID vaccine only they're asking.
Thyroid, but I don't think thyroid has any problem. That is nothing to do with the hormonal this thing, and it should be possible. But in any case, you ask specifically, Dr. Britt, as I understand, there is no such specific instruction. Thyroid is a hormonal imbalance. People need not take or something. It's not there. Only thing is, if you are having some specific problems, I told you diabetes. Even now, diabetic people are also advised to take. Earlier, they had some apprehension because as the experience is gaining. The doctors are asking people to take.
Any other questions, please? Any other questions, please? Okay. Okay. If there are no more questions, we'll give the vote of thanks after Kishan sir. Sir, last question.
Yes.
And regarding this COVID-19 pandemic, it is a natural. What is the reason behind to get this spreading of this one, sir? Because each and every pandemic diseases, they identify the various regions.
Yeah, it is basically say airborne disease to some extent. The reason is the people who have actually got this vaccinated, and when they say what's it called, cough and then exhale, the droplet infections, the fine particles which are present in the mouth or in the nose will go out, and when you take the same air, because of close proximity, so there is a chance that they enter through the nasal membrane, nasal mucosa into our body, and they spread. That's all. Only thing is, the best way is that social distancing. If you are away from others at least 1.5 meters to 2 meters, that is the best way. And second thing is taking warm water and then nasal drops or something like that. So these are all actually, as a matter of fact, in China, after this particular COVID say eruption in 2019, within 2-3 months, only whole Wuhan city, people used to take warm water and 3-4 times, and they have physical distancing and then mask putting. Completely they have control. They have not taken any vaccines, but they have control. It's actually, as a matter of fact, this vaccine or this virus is related to corona family. That is corona viridae family it is called. And there are so many flu viruses, etc. are all there related to that only. Somehow it has become so strong and it has got the spreadability like anything, and the people have not watched. And now the international movement of people from one country to the other country, and it has become increased. So naturally it has quickly spread, and people are not ready to say have any drugs, etc. for this particular virus. So therefore a lot of death was there.
Another final question, sir. Another one more question. And now the people are almost all, they're not following the guidelines given by the government. of India regarding the distance maintenance and mask and sanitizer, because they started the schools, colleges,
Yes.
and everything. And how to overcome this one? In spite of the so many
Because
times giving the instruction to the students.
And only thing to be at the moment, the reported death rate in our country for this COVID is less than 2%. That means if 100 persons got infection, let us say 100%, less than 2% have died. First of all, that is what exactly the problem. So since not large number of people have died, people are not taking it as a risk. That is one problem. That's the reason why they are simply working as careless. Because if everybody is in the neighborhood, somebody has died, then there is a big say wave of pandemic. The people get afraid and all. Nowadays, nobody. So that is the reason. And moreover, the hospitalization time has reduced, and some vaccines have also started coming, and then the medication, particularly say the repurposed drugs, to some extent reduce the hospitalization time, etc. Apart from that, Ayurveda. In Ayurveda, they have prescribed some say remedies, and that also giving relief. And we are not talking about the Ayurvedic relief and the nasal washings, and then the steam inhalation and gargling, and these are all some of the processes where the virus, if it is present and it is about to develop, spread, is controlled to a great extent. So these are all the things which are also playing a role in the lenience of the individuals and not seriousness of the individuals. But I don't know exactly in India, because it is something like a big country where large number of people are there, and they are not having the same type of attitude and nature to follow. That's it. Not only this vaccination, anything, even if you want to ask them to stand in the queue also, they don't stand. So they go this way, that way. That is human indiscipline is very common in this country.
Sir, another one, sir. How the hot water will be effect on this one, pandemic 19, sir? Because many doctors said that one. Hot water and either steam inhalation, both are is a very effective, they said.
Hot water, next one is?
And steam inhalation.
Okay.
It's turmeric.
Actually, the virus actually is having very susceptibility. The susceptibility of this virus is temperature susceptibility.
Okay.
If you take warm water, let us say, if some virus particles are here and there, they are going to be destroyed. Hot water, and then steam also, and the environment becomes not congenial for the spread and multiplication of the viruses.
Okay.
The environment becomes, because somewhat warmness means actually the body temperature will be slightly increased, maybe 1 or degrees, 2 degrees, not maybe 5-10 degrees, but this warm water or etc. If you inhale it, the virus is unable to withstand, but that small temperature is sufficient for it. It cannot spread. And washings, regular washings, that also dislodges from the place where they are going to be there. Hello, Dr. Gopinath.
Sir, okay, now okay, sir. Sir, the IS is the release that one, they said in the IS naturopathy system of medicine, and they give the clear instructions. If you do the pranayama, especially Bastrika and another Simmakriya, and enormously we can prevent the corona entry up to the individuals.
Yes.
Can you some more elaborate? Can you give that one?
No, because I'm not very good expert in this Ayush procedures, but I know that even I also feel and I practice also. Pranayama is going to help you a lot. That is one thing. And because ultimately what happens is that the lungs are the important organs which are going to be affected.
Okay.
And other organs are also going to be affected, no doubt. But lungs, important one is the respiration is going to be stopped. When you are continuously taking pranayama every day, the ability of the virus to infect and then cause a lot of distress, respiratory distress, etc., will be minimum. and moreover you are taking warm water and then say decoctions of particularly decoctions of this, I don't know, like this coriander or even jeera and things like that. There are suggested say decoctions, they stimulate them. The cells, they are going to be stimulated, and then the environment becomes not convenient for the viruses to survive there and propagate there and these things. So ultimately, the over a period of time, you will get washed up every virus and you are free, and your natural immunity is able to protect once again. So this is what exactly I personally feel. Not really good research, etc. I didn't do that, but I personally, my understanding is like that.
And okay, thank you very much, sir. For FDP program sponsored by AICT New Delhi and jointly organized by JNTUA University Anantapuram FDPSL and JNTUA OTPRI Pharmacy. They started with a very good startup and starting day, and the ending of the session also is a very good talk given by the Professor KISHAN sir, KU, and thank you very much, sir. And
so it's also my pleasure and to participate in this program, FDP program. Hopefully that the youngsters have taken some piece of information from my talk, and I'm also thankful to you particularly for thinking of me, and then thinking of me and associating myself in this particular FDP program, and I'm really thankful to you. And I thank all the teachers and other participants also for their time. And thank you one and all.
Thank you, sir. Shravani, please give the vote of thanks to the Professor KISHAN, sir.
Good evening, everyone. Finally, I take privilege to give oath of thanks for today's speaker, Dr. B. KISHAN Garu. Thank you, sir, for your valuable session on the topic vaccine delivery systems. Sir has introduced the vaccine delivery systems with a brief introduction. That is a common point: health is wealth, prevention is better than cure. So vaccines are nowadays more important in our life. And then the word vaccine has been derived from cowpox virus, and first vaccinated has been introduced by Edward Jenner for smallpox, and development of vaccine by Louis Pasteur, and then Sir has briefly explained about the traditional viral vaccines for human use and also bacterial vaccines, and then challenges for existing vaccines. Sir has also explained the new vaccine delivery system which have been required from the guidelines of WHO. And Sir has also explained the components of vaccine like adjuvants, advantages, mechanism, risk associated with these adjuvants, and also antigen, FDP approved aluminum salts, and the carrier for these vaccines, vehicles, emulsions, etc. And Sir has also been explained the proteosome and transfersomes, and then Sir has been explained about the nanoparticles for vaccine delivery to dendritic cells, and also explained the polymeric nanoparticles by the picture, and single dose vaccine delivery systems, and also oral mucosal vaccines. And finally, Sir has an delivery on DNA vaccine delivery systems, and then Sir has been explained about the coronavirus, which is an RNA-based virus and belonging to the Viridae family. And then first outbreak has been taken from the China and originated from the bats. And it feels very pleasure to present this oath of thanks for our today's speaker. And today's lecture was fully of knowledge and interesting things about the vaccine delivery system. It gives deep insight in the topic and also reveals some interesting facts about the vaccine delivery system. And the points where KISHAN Garu told about the vaccine delivery system was very informative. I am sure the previous precious knowledge that Professor KISHAN Garu gave us will definitely help us in our studies and future. Once again, I would like to thank Professor KISHAN sir for taking out time for this busy schedule and enlightening us with the knowledge. Thank you, sir. Thank you for your valuable session.
Thank you, madam, for your introduction as well as summing up of my lecture. Thank you, one and all.
Thank you, sir.
Sir, thank you, sir. Kishan, sir, thank you very much.
Bless you to everybody.
May God bless you.
May God bless you as well.
Thank you. Thank you, sir.